So today, in our ongoing Coding and Vision lecture series, I'm going to talk about brain imaging, particularly functional brain imaging, something we don't do at the Institute, although we work with people who do DTI, it's one form of imaging techniques, and structural MRI and functional MRI. So this is a picture from Alan's TED Talks. It's a beautiful brain here at the Institute, very red, lush. And it looks very homogeneous. So since antiquity, people have always distinguished gray matter from white matter, but it's only in the 1800s that people first saw the striae of Gennari, the termination of the axons in the visual cortex. Before it was pretty featureless. And then, of course, only subsequently with the development of chemical dyes and the co-development of the chemical industry that made those chemical dyes available, that people could begin to differentiate cortex and the other organ. The birth of neuroscience, I think one of the founding fathers of neuroscience is not frequently recognized, is his German physician, Franz Josef Gall, who, of course, who operated he wrote his most important book in the early part of the 19th century, where he made these statements that at the time had to force him to flee. He was living in Germany and in Vienna, but because these were profoundly against the spirit of the time, they were conceived of profoundly anti-religious, he had to finally emigrate and live this last year in France. So made these statements that to us are obvious, but he defended them at the time when they were not popular, when there was little evidence. The brain is the organ of the mind. The brain is not homogeneous, but an aggregate of mental organs with specific functions. So they're very specific. They're discrete functions done by the mind, and they have discrete localization in specific parts of the brain. That these parts in the cerebellum, in the cortex, are topographically organized. So with nearby regions doing nearby things, similar things. That the relative size of any one organ and it's indicative of the power, the strength of that organ. So the larger the cortical area dedicated to a particular task, the more important that task is for that specific individual. So we're trying to link structure with function, and this we find today. Of course, we don't think this is true anymore, or we know it's not true. So since the skull ossifies over the brain during early development in childhood, external measures can be used to diagnose internal states. So on the one hand, this gave birth to the science or the pseudoscience of phrenology. On the other hand, it was an early attempt to generate biomarkers, right? Because the claim is that you can measure the skull, and you can sort of, there were specialists who did this, you brought these in to view the skull of your children and tell you what their strengths and weaknesses are. Depending on, if an area was particularly well developed, the assumption was that the bone over there is sort of thicker, and there's a bump there, and you can infer something about the underlying characteristic of the mental organ of that particular individual. So today we mainly make fun of that idea. We know that's not true. There really isn't any relationship, except in the most extreme cases, between the outside skull and the underlying brain. But at the time, it was a thoroughly modern idea. And there's still, interesting, our language still retains some of these elements, like highbrow, right? When you say that, well, that's a highbrow form of culture. Opera, for instance, often said is highbrow. Well, there was meant somebody, intellectuals and deep thinkers have a high brow. So, as I said, this gave rise to this enormously popular, but at the time it was attacked by many scientists, but it was still very popular, phrenology. And you can all see these statues today. And what's most striking about these, if you look at actually the detail of these atlases, they don't pertain at all to low-level features like color or motion perception or audition. It's always high-level things like love of country and love of self and things like that, rather than sort of the more elementary psychological modules that we might think of as today. Like I said, they had one color, but it was up here, so all the details were completely wrong. And we can fast forward in the attempt to try to understand cortex. And this is really the birth of functional neurology, which took place in the middle of the 19th century in France, at the time one of the most advanced places to do brain science, by this French physician, Pierre-Paul Broca, after which, of course, we named Broca's area. And he studied his first and best known patient. He's called TAN in the literature because the only thing due to a stroke, the only thing this gentleman could utter was TAN. That's the only thing he could say. He was had a severe expressive aphasia. And he died a couple of days after Broca first investigated him. And based on this evidence and one other patient that had a similar lesion here that was also aphasic, Broca made the claim at the time that was readily accepted that he did a structure-function relationship, that this clearly indicated that particular parts of the brain were responsible for particular function. In this case, language area is done by this area in the inferior frontal gyrus, IFG. And so today that's this area in his honor is named Broca. This was very, very important. And for a long time, for the next 30 years, this was really the only area that was localized. Only later, and towards the end of the 19th century, that people localized vision to the into the back of to the occipital lobes. So this was the first structure-function relationship that people studied. Of course, interesting, there's a very strong usually left asymmetry. Most people have it on the left, some people have it on the right, and a small fraction of people have it on both sides. Interesting, they rescanned, this is a paper in 2007 where they rescanned the two brains, they're preserved, and actually found that there are actually additional lesions. So the patient, it's not a pure cortical lesion, there are also other structures that are implicated. And here we are 150 years later. So 150 years later, based on the cumulative efforts of thousands of neurologists who write papers about their patients, we know much more. In particular, of course, today the name of the game is always to find very, very specific lesions that are as specific as possible, because it's not interesting if most of my brain is gone, then of course it's not surprising that I can't see anymore. But if I have a fully intact visual brain, but I have, in one case, was published in Neuron a couple of years ago, I only have a deficit. I cannot see color in the upper left quadrant. So I see normal color here, here, and here. In fact, the patient wasn't even aware of this specific deficit, and it's only after detailed testing that Jack Galland at Berkeley realized that this patient has loss of color vision in the upper part of his left quadrant. And in fact, if you scan the patient, there's a very small lesion in the occipital temporal area. And so here we have this is sort of a cumulative diagram from a textbook. So this is the neurologist convention. Neurologists used to look at mainly at dead people. So you look at them from below, the feet sticking out. So on the right side of the image is the left brain. So you can see here. This is achromatopsia, so this is color blindness. So it's not the one, it's not a retinal color blindness where you only have two genes, that's mainly in man, but this is if you have destruction of you have normal color vision, but then you lose either this or this part of the brain, and then you have these color deficits I was just talking about. Prosopagnosia, also known as face blindness, as in Oliver Sacks' very famous story, the man who took his wife for his hat. Achinotopsia, very rare, it's really only two patients, one in World War I and one in the 80s, where typically you have to have bilateral lesion in order to have loss of color vision, in which case the world looks like in a disco. Right? So when this, particularly the last patient, which was quite well described, the patient, she sees a car far away, and then it's close by, then it's here. She see, just like you see discrete snapshots. The patient's unable to perceive smooth motion. And then alexia, inability to read. When you read a newspaper, it suddenly doesn't make any sense anymore. Again, this is lateralized to the left hemisphere. The other one tend to be bilateral, although with prosopagnosia there tends to be a right bias. Anyhow, so based on this evidence from the clinic, we can make very specific structure-function relationship in assignments in cortex. Then people, as I mentioned early on, the development of the chemical industry, particularly in Germany, gave rise to a myriad of different stains where people can differentiate different cortical tissue. This is the most famous one that we still use today. by Corbinian Brodmann, a German neurologist. So then at the time already, this is now the 19th century when he was active, you had this differentiation from just being a mere physician as Gall was or as Broca was to specialist, brain specialist. This is the first type of brain specialist. Of course, today there are many. So he made, of course, famously a map of the human brain. He identified on the order of 50 different areas and basically named them contiguously. Left out one or two because he found them in monkeys but not in humans. So typically there, it goes up to 52. This is primary visual cortex area 17, and over here 44, 45, that would be Broca's area. This is based on minute, these people were very, very good observers, and they found minute differences in the texture, the way the brain looks under microscope, under these stains. And once again, they were working on the assumption that there's a close relationship between structure and function, that if you see minute differences in the structure, let's say the density of something is different, or it looks a little bit more brownish, and over here it looks a little bit more reddish, that probably reflects some difference in the underlying function. And so far, that's always been proven to be true. Now today, of course, we use much more finely distinction. So for example, Brodmann's area 18 is today subdivided into V2 and V3 and V3A and many more areas. But remarkably enough, this is still used today you really did this 1909, yeah, so I guess 100 years later, it's still used today. Think of it like a zip code, right? It's a crude orientation where things are in the brain. Of course, it's not always accurate. There are some boundaries which are very accurate, like the 17-18 area, but others are sort of more, with invaginations and other inhomogeneities. But it gives you a rough feeling to where things are. Then we can fast forward roughly 100 years by this pair of an American scientist and 40 of their colleagues, Michael H. and Ed Lean. What I wanted to show you, so this is a map, the matrix representation of differential mRNA expression for two human brains. They've now subsequently done it for two more. So each entry here, so if you take one particular entry here that says if you look at the number of differential neurons, genes expressed between V1 and let's see here the temporal pole, it's, what is this number here, 50. Blue means a very small number, and brown and red means a very high number. So what this tells you, if you look at the one end here, the cerebellum, so the cerebellum within, there are not many genes that are differentially expressed. Same thing with a few exceptions, like particular primary visual cortex. In general, in cortex itself, there are not too many genes that are differentially expressed, or not nearly as many as in other structures, like, for example, the hippocampus or the striatum. So in other words, it's relatively homogeneous. Now, of course, one can see very consistent differences in gene expression, but they tend to be much smaller and much fewer than in some of these other areas. So this supports sort of this argument that cortex is basically a homogeneous structure with lots of specialization, but it's much more homogeneous than most of the other parts of the brain. Of course, it's also area-wise the biggest part of the brain, and the part of the brain that we think most distinguishes us from other animals, like monkeys and dogs and our other close cousins. Also, what I find so terribly intriguing about this data that they tried very hard, but they could not find any left-right asymmetry, any statistically significant left-right asymmetry. So in other words, we know from Broca's data and other neurological data that there's a profound asymmetry when it comes to reading, speaking, and writing. Those are all typically left-located. Yet if you look at the gene expression, at least at this averaging over the entire gyrus, you don't find a difference between the left part of the brain and the right part of the brain. I find that very striking. All right, now this work, of course, was always done on either injured people, like the patient from the clinic, or on dead people, on dead tissue. Now, it would be wonderful if one could look at living tissue in a breathing and behaving person and do it safely and routinely and repetitively. And this is what this technique called magnetic resonance imaging has enabled us. It's fundamentally changed the way neurologists operate. It used to be until the 80s or so, the main one big job of the neurologist was to locate exactly in the brain from the outside, without using these imaging tools, to locate where, for example, a tumor was. Today that's trivial. That's not really the job anymore because that's, it's very easy to do with MRI. And of course it's profoundly changed psychology and given rise to this entire field of cognitive psychology. And you can now go, if you go to the annual meeting of the Society of Cognitive Neuroscience, it's essentially 90% plus uses this technique fMRI, which is a particular type of magnetic resonance imaging. So it's a tool to safely routinely image soft tissue. It's almost entirely used to image protons, hydrogen, because most of the brain is water, so it's very easy to image that. You can also do spectroscopy, where you image the presence of other nuclei like C13, etc., but that's not done. So it's great for imaging brain, also the heart, ligaments, tumors, things like that. Spatial resolution depends on the tool and the instrument, but typically it's around 1 to 2 millimeters. So the highest resolution right now for humans is 1 by 1 by 1.5 millimeter. In animals like in macaque monkeys, where people also do imaging, when you implant the coils into their skull and you can leave the monkey inside there for 8 hours, then you can get resolution approaching a quarter of a millimeter. But that's of course not possible for humans. It was developed in the early 70s, so the technique isn't all that old, by Paul Lauterburg of Illinois, Peter Mansfield in England, and Raymond Damadian. These two gentlemen got a Nobel Prize for 2004. He didn't, and you might have, I don't know if you remember at the time, there were big, he and his friends got together and wrote big advertisings in the New York Times that this was very unjust, and there was a big controversy over it. But I mean, those are the three individuals that are typically mentioned in connection with MRI imaging. And so you can have these wonderful images today here. So this is a typical X-ray CT image, which is not very useful if you actually want to image soft tissue. It's great and continues to be in use, of course, if you have a broken skull or broken ribs, but it's not very useful for imaging details. And this is a T1 weighted MRI image, which is very high resolution. We've all gotten used to it, but it's really a miracle if you think about it. Essentially you bombard the brain with radio waves, and then you get these fantastic images back of a living, breathing individual. So what is NMR? NMR is a physical phenomena in which nuclei, not electrons. Electrons also have spin, and there's something called electromagnetic resonance. But in which nuclei, like hydrogen or deuterium or any other nuclei that have spin, where the spin, this quantum number is different from 0, in a magnetic field they align, and if you bombard them with particular types of electromagnetic radiation, at a particular radiation, they'll absorb a photon and then can send it back. And so this radiation is known, this particular frequency is, at that frequency they exhibit what's called a resonance. It's very sharp. It's the one particular frequency that you have to hit, which tends to lie for things that we care about as scientists, they tend to lie between 30 megahertz and 300 megahertz. So it's really in the FM radio. It's like listening essentially to it's bombarding your brain with radio waves, with FM radio waves. Right? So this is an entirely quantum mechanical properties. It cannot be explained by classical physics. It was discovered in the 1920s that all nucleons, i.e., all nucleons, protons and neutrons, as well as electrons, these are elementary particles, that they have this thing called spin. Now, if they were big-sized particles, you can think that they have an axis of rotation, they spin around there. Now, an electron is an infinite, as far as you can tell, infinitesimal particle, so you can't really think of it. It's not really a spinning, but the best way, with our limited imagination, think of it just like a top and that spins. Right, so they have this property. We didn't know about it till 1927 or something like that, but it turns out it's one of the best, in fact, it's the best verified physical property known to science. People can measure the magnetic moment of the electron to a staggering, they can measure to a staggering 10-digit accuracy, and then they can calculate it theoretically from quantum electrodynamics. The difference between the calculated number and the estimate and the actually measured number is 10 to the minus 10. That's like if I predict what the distance between Seattle and and York is, and I can get it right within 1 millimeter. I mean, that's a sort of the precision. So it's by far the best number we know in science today. So it's a physical property. You put these electrons, and you put these nuclei that have a spin. Not all nuclei have a spin. It depends on how many neutrons and protons they have. But H1, for example, has a spin of 1/2. So in a magnetic field, they all align. So you apply a strong magnetic field, and then the nuclei will align. And then they precess around the axis of the magnetic field. Now, a nucleus like hydrogen with a spin of 1/2 can take actually two states. It can be oriented parallel to the magnetic field or anti-parallel to the magnetic field. And there turns out there's a slight excess, a tiny fraction of a percent excess of protons that are aligned with the field, parallel with the field, than anti-parallel with the field. Right? And now what happens in the presence of a magnetic so you apply a magnetic field, and they tend to align here, and so they tend to precess around that field, either like this or like this. And there are a few more that are parallel, just a tiny number. I mean, I couldn't draw it here because it's 10 to the minus 3 or 10 to the minus 4, but that's sufficient. You know, there are a lot of the 10 to the 23 per mole, and there are a lot of these, okay? And now what you do, if you apply a magnetic field, you get what's called the Zeeman effect in physics. You get the hydrogen nuclei go into two different states. They go into a state where they have into the S, where they oriented parallel to the field, and they go into a state where they oriented anti-parallel to the field. And the difference between these two is sort of delta H, and the difference, and delta H is directly proportional to the applied magnetic field. Now, of course, you can think of this as the energy is just h over 2 pi, right? The Planck's constant times frequency. And so you can think of it, what happens now if you bombard this with radio frequency that have exactly this frequency here of gamma times B. B is a magnetic field. Then what happens at the right frequency, so it depends on your magnetic field, at the right frequency, let's say here you're at this field, and if you have this right frequency, and the frequency would be expressed by this relationship, then the protons will absorb energy from the radio wave and will go from the low energy into the high energy state. And then if you turn off that radio wave, after some time it'll flip back. Right, so what you do, you put in a pulse of the right frequency, and they go from the low energy state, let's see, the anti-parallel to the parallel state, and then you turn it off, and then they'll essentially relax back. And what you're measuring now, essentially, what relaxing back means that they radiate, when they make the transition from the one to the other state, they'll radiate back that particular radio frequency, and you can pick that up. That's essentially what you're measuring. And the decay has different time constants. You can sort of depends how quickly this signal decays, depends on all sorts of things. It depends, for example, of the exact chemical environment. Are there electrons around it, and do they filter the nucleus, or do they not filter the nucleus? And it depends on the interaction of this one H to other H's around it. And so it's a very sensitive indicator of the local magnetic environment, that changes, for example, with the concentration of protons, i.e. water. And it also changes fortuitously, for example, between hemoglobin and hemoglobin. When you have, I'll talk about that in a second, when you have blood flow that either carries oxygen or doesn't carry oxygen, the magnetic property also changes. And so you're sensitive to all of that. So what you do now in MRI imaging, okay, so the basic technique is you have a radio frequency that's tuned to this. It's called the Larmor frequency, the frequency at which this nucleus precesses. If you obey that relationship I just showed you before, it resonates. So that's why it's called nuclear magnetic resonance here. The radio wave photons absorbed by the H1, and the proton it's in its energetic favored state. You turn the EM field off, then it returns to thermodynamic equilibrium, and I think I have a demonstration of this here. So this is what you can actually measure this as radio frequency. So this is hundreds of milliseconds here, and this time constant here is called T2 prime. I mean, for the experts, T2 star. Essentially, it's a rate at which the H1 return to the equilibrium. That depends on all sorts of things like the spin density and the chemical environment. So that's the basic thing, the basic physical phenomena that you're exploiting in NMR. What you do now, you establish gradients of the magnetic field, so you can systematically. So each point has each voxel in the brain can be addressed using a specific, slightly different magnetic field, right? So you know where you are, and you do these gradients in both x and in the y direction. And this is one of the technical challenges. You want a very homogeneous field, but you also want a field that has a strong a gradient. So like I said, so each voxel has their own magnetic field with their own resonance frequency, so you can identify from which location you're receiving the signal back from. This was really Mansfield's doing. And then what you get, so this is my brain imaged in a high field magnet in Zurich. So then you can get these beautiful images that we all used to. So by the way, here's primary visual cortex. Here you can see the bump called the ilium. You all have a bump here at the back. You can all feel it. Try it. There's a bump here at the back. It's called the ilium. It's this one here. So your visual cortex just attached just a centimeter above there. And here you can see the parietal occipital sulcus. Here you can see the LGN, left and right LGN. It's all there. First time you worry when you see your brain. And you can also do, okay, so this is done by Mike Tischka at Caltech. So here he just did this continuously, I think every 300 micrometer, you can get these beautiful movies, high resolution movies of a volunteer, right? This is completely safe, this technique. The only thing that you can get at high field strength, if you go, for example, into 40 magnet, or there's now a couple of 70 magnet in the world, that if you move your head very quickly, you can sort of see flashes of lights or things like that. That's sort of due to magnetic induction. Sometimes people get a very brief feeling of a passing feeling of nausea if you move very quickly. But otherwise, it's completely safe. Unlike, for example, PET, which is also a functional technique, but which you can only do a very limited number of times. This is, I like different imaging techniques and I like to apply them to my own body. This is the same thing, MR imaging techniques, but here it's applied to the heart. So here, because you have these huge blood volumes, of course, so this is just 2 heartbeats. You can see sort of 2 heartbeats, the heart going through 2 heartbeats. And it's, I don't know, I find it an amazing technique. You lie there and you can see your heart beating in real time. It's on my website together with the U2 song, Slow Down My Beating Heart. All right, so this is all, well, except the heart. This is structural imaging. So structural imaging is fantastic. And for example, one of the descendants of this technology today is called DTI, diffusion tensor imaging. Now, let's move to functional imaging, because we'd like to image the brain when it does something, and then we can infer about where that function is maybe located. So the first, as far as I can tell, the first paper to do this in a human came out of MGH in Boston, in Cambridge, about 20 years ago. And you can see here, so this used a contrast agent. This used a contrast agent, so you were injected with gadolinium to enhance the signal. And you can see here, just like my brain before, here's cerebellum, here's a visual cortex at the back, and they sort of took this plane through here, and you can see it. So this is when the person was in the dark, and this was when the person was in the light. So it's a very, very, very simple experiment, but it's the first time they could image this, just essentially light and dark. And then here they did the differential, differential imaging. And so you can see, just using a simple subtraction technique, you get here signals in sort of visual cortex due to the fact that there was input. So it just shows what we knew already, but it demonstrates for the first time the potential of this technique. Now this has a drawback that you need to use this contrast agent, and that limits the technique. Now it turns out there's a fortuitous link. In fact, you can see this yourself. So if you look at your blood system, you realize that there are 2 sorts of there are arteries that are deep. Usually you don't see them unless they're cut, and then there are veins that are more superficial that are bluish, right? So we know already just from looking at our bodies or from our common experience that, that blood can be two different types. We know one is fully saturated, the hemoglobin is saturated with oxygen, then it looks nice red. And then when it's deoxygenated, when the oxygen has been given off, it looks different. Well, it does not only have different optical properties, but it also has different magnetic properties. And that's essentially the reason we can do fMRI, because essentially we have this internal contrast agent in our heads. 1 year later, 3 papers appeared simultaneously. So fMRI really dates from 1992, sort of the fMRI that's mainly used today by most people. And so it exploited simply the fact that deoxyhemoglobin is paramagnetic. So essentially because you have these unshielded ion at the heart of the hemoglobin molecule, well, oxyhemoglobin is, because it's bound, the oxygen is bound, is sort of only very weakly magnetic. It's a so-called diamagnetic. And this is a property that was discovered by Linus Pauling. So many things were discovered by him at Caltech 50 years earlier. So he measured this. It's a very simple measurement. He measured that hemoglobin and oxyhemoglobin have different magnetic properties. So it's something that was well known and people looked for. And this is the basis of the most popular imaging techniques, variant of MRI imaging techniques called blood oxygenation level dependent fMRI, so BOLD. So that's what people talk about. BOLD imaging. This is the technique they use. So the N was dropped in early days. It used to be also known as NMR imaging, but people didn't like the implication of nuclear, right? Nuclear bomb, nuclear age. So they just dropped the N. And of course, today, 20 years later, and I Googled, checked 20,000 papers just published with this one technique. So now, right now, people publish in fMRI at the rate of 10 papers a day. So each and every day, there's 10 papers that appear somewhere in the literature that use fMRI. So this is, for example, a standard. This is the one we have at Caltech. Well, we have a number of them, but this is one of them, sort of the workhorse. Typically, many departments will have them by GE or by Philips or by Siemens or by Bruker. Those are the 4 major companies that make these machines. Now, they're based, so once again, the underlying, what you're measuring is differential blood flow. And so it turns out the brain is incredibly precisely supplied. It's not that surprising if you think about it. It precisely controls its blood flow. So this is a technique. This is from the Logotitis lab, where they inject 2 agents into the blood system, and if they combine, they rapidly turn into plastic. It's called plasticizer brain. And then you can etch all the other tissue away. And then what you get are these beautiful casts where all you have, you can see sort of you have plasticized now the vascular system. So this is cortex. This is the top of cortex. So you get these very big arteries. And then here you get they branch into ever more finer arteries until finally here you get the capillary bed, and the smallest one. And then from the capillary bed you end up in the draining veins. You can see this. Now remember, all the neurons have to live in here too, right? I mean, we don't think about that. We always think about neural tissue. Well, I mean, neural tissue, you don't get, every piece of the brain has to be within 100 micrometer or so from a capillary to supply its oxygen, because otherwise it's not going to be able to live and function. Right? So that's what you have when you get anoxia, when you get a little stroke. The tissue dies unless it's supplied within seconds by blood. We know this because if you choke somebody, the average time it takes, people have measured this in, quote, Navy recruits in the 40s. They choked a couple of hundred recruits, and they found that the average healthy Navy male recruit takes 8 seconds to faint. So we don't have a lot of blood supplied extra. And yeah, so one should think about the same thing with the mouse, that all the neural mechanism is that we only focus on has to be supplied by this capillary bed. Here it's at high resolution, very, very beautiful. I mean, look at how, so this is 100 micrometer. Look at how tiny these things are. And here they have this sort of active regulation. It's all these mechanisms that people discover now to regulate the blood flow, because ultimately what we're measuring, so it's differential blood flow. That's essentially what we're measuring. We're picking up hemodynamics. We're not measuring neural activity. I'll talk about it in a second, but we're measuring blood flow. So the signal can only be as precise as the blood flow is regulated in the brain. If it's very precisely regulated, at least leaving aside physics, we have a hope, at least in principle, of being able to measure as precise as the blood is regulated. So it turns out that these precise mechanisms, turns out there are these interconnections between astrocytes, between certain types of interneurons, and astrocytes that then wrap their processes, like here, around these tiny capillaries to constrict them or to open them. So to really differentiate. So the idea of any functional imaging is that if I look at something, let's say, colorful, and I have a bunch of neurons that are particularly active when there's a colorful stimuli, then the blood will be sort of the hemodynamic flow will be enhanced to that particular location to compensate for the power consumption. All right, so you can do this routine experiments done a couple of years ago in my lab. This is total routine, one of the best area that can be imaged in anybody. So this is primary visual cortex. Here you have this area called MT, sometimes also known as V5 or MT+. It's bilateral. This is the area that if you lose it on both sides, you get ekinetopsia. Just like this lady I mentioned earlier, she's unable to see continuous motion. She sees this chopped motion. And so this is the stimulus you use. You use these dots that expand and contract. It's a very strong stimulus. And then you get fMRI activity both here in V1. On a time scale, or you can see here the time scale. So this is many seconds. It's sluggish. It's not sluggish due to the physics. It's sluggish because you're picking up blood flow. You're not picking up the underlying neurons. And then what people do, they have this technique I'll show it in a second, where because it's very difficult to just look at the brain, given that it's highly folded. So what they do, essentially, they take a pump to it, and they pump it all up, just like you have a tire, and it's all creaking. You put air in it, and it becomes nice and round. And so here you can see it on this so-called inflated brain, where these different parts, where this area MT is. And we know in a monkey, okay, I'll come to that in a second, a monkey that's filled with neurons selective to motion. So now this is now based on fMRI. Before I showed you a picture like this based on neurology, when they infer you have a patient with a specific lesion and a specific inability to read or inability to see colors or something like that. This is now in intact humans by Nancy Canwisher, a review paper of hers. So the implications are slightly different. So based on the lesion study, we can say these areas are really essential, because if you lose that area, you get phase blindness, for instance. So one of the best explored areas was discovered by Nancy Canwisher, called the fusiform phase area. Here it's just the fusiform gyrus, sort of above the cerebellum. In fact, there are several of these areas. Once again, this is left and right is here. So they're particularly represented on the right hemisphere in everybody. These are areas that get specifically activated much more strongly for phases than for most other things. So so people like her distinguish now between regional specificity and functional specificity. So regional specificity is where you have some function, let's say motion processing or phase processing, or places. When you look at, for example, buildings or mountains or things like that, there's a bunch of neurons here in the parahippocampal place area that get preferentially activated. So you have regional specificity where some function activates region X more than other cortical regions, versus functional specificity. Region X is more engaged for function Y than for other functions. Because it's one thing we should never forget, right? When people show these maps here, for example, for phases here, it's not that these are the only places in the brain that respond to phases. If you show phases to people, the entire visual brain will light up, because, for example, V1 will, of course, respond to phases, and V2 will respond to phases. If it moves a little bit, MT will respond to phases. Right, so lots of things will respond to phases. The implication is rather that these areas are specifically activated by any task that involves phase processing. So conversely, the claim would be that if you lose this area here, or maybe in some cases bilateral, you would then get phase blindness. And in at least one patient with a very precise lesion, that's been the case. So people find other areas. There's one again that corresponds to sort of the twin to alexia, right? So alexia is the inability to read, and indeed here on the in the left hemisphere, you find an area that responds specifically to visual words, so the so-called visual word form area. You know, we read hundreds, you know, I don't know how many hours on our computers or in books or newspapers, we read things, and so it turns out there's a part of the brain that's particularly dedicated to that, again, on the left hemisphere. There are other areas that are dedicated to when I make inferences about what other people think relating to theory of mind, there's an area that responds particularly to bodies, etc. Here, so this is a slide from Stander Hayne, one of the people who discovered this visual word form area. Again, on the left side, that specifically responds to words, and typically it responds much stronger to real words rather than just a random matched list of consonants. So if you just have a random 6 letters that don't mean anything, this area will be activated, but it'll be much more strongly activated if you actually present real words. And Canwisher, in fact, from her lab, we also know that this area depends on experience. It makes perfect sense, right? We only have language a couple of thousand years, so it's unlikely that there's a genetic, that there's a gene for it specifically. So here you have a bunch of MIT undergrads, none of which is a Hebrew reader. And you can see, so this area is strongly activated by regular text and by words, and also by numbers, but much less by Hebrew, which shares, of course, some of the primitives of individual letters. And here it's 11 undergrads that are Hebrew readers, and here you can get the signal is much, much bigger now. It makes sense. It depends on experience. So, some of the many things you can infer using fMRI. You can also get to very subnucleus in terms of regional specificities. The paper came out last year from my lab. This is based on work that we're doing. This is single cell human with Itzhak Fried, where we find a preponderance of cells in the right amygdala, single neurons that respond to animals, and then if you put normal subjects now into a magnet, you find the same, that you get activity in the right amygdala, but not in the left amygdala. All right, now I wanted to talk a little bit about maps. So before I'm just talking about location, right? You can try to isolate specific functions and to find their counterpart in cortex. Very similar to what Gall said 200 years ago, essentially, what he predicted would be true. Now, what people are very good at, particularly in the visual case, of mapping out. So there's this topographic relationship between the exterior world and the brain. What I mean by that two nearby points out there in visual space, map onto nearby places in visual cortex. And points that are far away will map onto far away points in visual cortex. That's called a topography. I mean, that's the very definition of a map. It's somewhat distorted. It's distorted in a peculiar form, peculiar to us, primates. It's not the case in monkeys, i.e. the fovea, the most central part of our vision, is greatly enhanced. So more than half of our visual brain is given over to the analysis of the central couple of degrees. So my thumb at arm's length is roughly 1.5 degrees. And so that part, I think, is 20% of my cortex is just given over with that central part. That's why I move my eyes all the time, just like you do. I move my eyes 3 to 4 times a second because I want to bring the high resolution portion of my eye over whatever I'm currently interested in. And that's expanded also in the brain. That's called a retinotopic representation. That's peculiar to primates. So people have imaged that. So as I said, this is the left hemisphere viewed from the back, and then, as I mentioned, there's this technique that people developed in the late 80s when they inflate or unfold cortex, where the black and white means the gyri and the sulci. And then they can exploit the property that the brain has this topographic representation. So here, outside in the world, you're looking at this grid that expands. And you can see here, really cool, the traveling wave of hemodynamic activity that we think underlying is, and I'll talk about that in a second, is a neural wave. But for now, all we can see is this hemodynamic wave. Very, very nice, very lawful, very systematic. And then there's also a technique, I don't show it here, where you can do the same thing with angular mapping, where you sort of move a wedge around and you can see the counterpart moving on the surface of cortex. Of course, you can only see this wave after you unfold it, right? That's one reason why you want to unfold cortex. So here you can see the different representation. So here's a primary visual cortex. Here's the horizontal meridian, V1. So here's the borders of it, the outer sides. And then, of course, they go continuously into V2, right? There's no discontinuity here. And at the brain level, it maps. If you go from the depths of the calcium sulcus to the canyon wall, to the lip of the canyon right outside, it goes into V2. It ends here, and then this is where V2 representation starts, and the V2 representation moves to the center, and then over here it reverses again. So people have done this very nice mapping. And here you can see the eccentricity mapping you saw before in the movie. So the fovea is mapped here, and the more peripheral parts are mapped here. People have described this mathematically. This is known as a log mapping. And you can see here the angle mapping. So in general, the upper part of the visual field maps onto the lower part of the calcium sulcus, and the lower part maps onto the upper part, on the upper lip of the sulcus. I think of it like a canyon. And of course, left maps on right, and right onto left. So we really know very well this mapping, and you can study it, for example, in people who are albinos, who have, of course, a different decompensation than normal subjects have. And there's these very nice, particularly Wandel at Stanford has done this very nice lawful relationship between the structure of the eye and the optical pathway and the fMRI signals here in the back of the brain. So these are among the best understood functional imaging examples, partly because the signals are very big. In general, the fMRI signal is small. You'll see it. We're talking about a change of 1% or 1.5% above the baseline. In more central area, when you try to do imaging in the hippocampus, the signal might be as low as 0.1% or 0.05%. Attentional effects may be a factor 20 there. So we are talking about very small signal to noise. So a lot of the frontal lobe is bedeviled by the fact that the frontal lobe, medial temporal lobe imaging, the signals are small. Partly you get distortions due to the presence of the ventricles that are filled with air. So what people have done, they've derived these maps in human. So this is the inflated human brain. The back part here is V1, the upper and the lower part. So you can see this bevy of different visual areas, V1, V2, V3, V3A, V4D, and all of that. So there are at least 12 of these very topographic areas here in the back. This is the counterpart in monkeys by either David van Essen or Leslie Angerleider. Now what's interesting here, that these maps were established based on single cells. This is sort of work done in the 70s and 80s in single cells. And here people find homologs in humans. You can clearly see the evolutionary relationship. They're not exactly the same, but they're clearly related.
What's the size of a cortical column in relation to this?
Oh, okay, so this is, there's no scale by here. This is, of course, bigger. In us, it can vary by a factor of 3 between 1,000 square centimeters and 2,500 square centimeters, while here it's considerably smaller, so this must be factor 2 or 3 difference here. I didn't show that. It's interesting, there's a lot of variability in humans. People have measured this. I mean, they've noted this, measured it. There's a nice paper that does postmortem anatomically in V1, and people have also done it functionally, as I show in the next slide. There's a big factor 2 to 3 difference in the size of primary visual cortex. And in fact, this shows up in perception. Once again, this is something that Gall had predicted. Remember when he said that the size of the organ has to relate to the, in cortex, has to relate to the importance of the function. Well, here what Geraint Ries did, he's an fMRI, one of the best fMRI imager at the Wellcome in London. He looked at the relationship between the strength of a visual illusion and the size of cortex. So here this is a so-called Ebbinghaus illusion, one of the classical illusions discovered in the 19th century. So if you just look at things, this disk looks much smaller than this one, right? Perceptually. Well, of course, you know by the entire frame of this context that, of course, it's not. If I were to cover this up, you would see that these are exactly the same physical size. It's just that relative to these large surrounding ones, this one looks smaller, and relative to the small one, this one looks much larger. Now, you can measure the strength of illusion. You can ask people to objectively, for example, to match, they're given a disk and they have to match, match this disk over here to this one just by changing the diameter. So there's an objective way how you can measure the size of the illusion. And different people, as people want to, will have different sizes. And you can do this for all sorts of, they also did this for the Ponzo illusions or for other illusions. And then you can go and measure different sizes of the visual brain. So you can use fMRI to identify V1 or V2 or V3 in three volunteers here. So you can see here it's 1,600 square millimeters. Sorry, did I say centimeter? Yeah, square millimeter. 1,400 square millimeter and 900 square millimeters. And you can see there's a systematic relationship. Here the brain is much bigger, the illusion is much smaller. Here the brain is much, much smaller, 900, and the illusion is much stronger. This is not true for V2 or V3. So people, you can now go, why should this be the case? So people have now found a number of these structure-function relationships where they try to correlate something, the way individual people perceive something, with a structural feature or functional feature of their brain. Now, to end, what are we actually measuring? What's actually measuring here? It's magic still at this case. We have this fantastic technique. You can see the brain in action at high resolution, but it's not clear what is actually being picked up. There's this complicated cascade of signals that originates with neurons. So the first thing, just because of the speed, I open my eyes, I see something. Within 50 milliseconds, my neurons in primary visual cortex will be activated. Within 200 milliseconds, most of the neurons in most of my brain will be activated. That's just a trigger for a cascade of events. Neurons are activated, synapses are activated, right? They're very expensive metabolically, a lot of ATP that needs to be used, they need to be recycled, glucose mechanism, so you get all this glucose and oxygen metabolism activated. That turns on a cascade of events that changes the blood flow, the oxygenation level, of course, changes, and the volume of the blood flow. All of those things change concurrently in a very complicated way that people are only now people are now untangling in quantitative ways. So people are now beginning to model this in very quantitative ways. So those are the physiological effects that we are seeing. This will then impact the magnetic field uniformity, and this essentially is what's being indirect measured by the NMR effect. This changes the decay time, and that's actually, then you get this T2, this decay time weighted image intensity, and that's finally what you analyze and what you show in color in your paper. So it's a complicated series of effects, and what's not clear, what's the trigger here? Is that increase in firing rate? Is it increase in firing rate in excitatory cells or inhibitory cells? Is it synaptic activity? So there are a number of attempts over the last 10 years to answer that. So one of the first one came with Geraint Ries, the guy I just mentioned, was a postdoc with me. So what he did, as I mentioned before, area MT is one of the easiest areas to see in everybody. You get it very routinely in every single subject. So here you have three different motion signals, a very standard classical task developed by psychologists and by animal physiologists. You have a motion signal that either moves up, either up or down. Here it's totally random. Here 20% of the signals are moving, I think, down. And here 100% of the signals are moving down. So what people like about it, it's a very quantitative way how you can study motion. It's the same dot, same density. The only difference here, every consecutive time they get remapped randomly. Here 1 out of 5 dots gets remapped in this direction. Here every dot gets matched in that direction. So you can systematically vary this. So people first did this, Bill Newsom in particular, at Stanford, or Stony Brook, and at Stanford did this in a monkey. So you identify area MT. You can see area MT in a human and monkey because it has very high myelin. You can see it in a myelin stain, because it's dedicated to processing motion signal. And so they need to get there as quickly as can. That's why there's this high degree of myelinization. And what they found in the big neurons, well, they found in neurons that they're recording from. Now, of course, given the bias of the electrodes, those tend to be big neurons. They find that this relationship between the coherency of the stimulus, remember 0, dots moved randomly, 100%, every dot moved in one direction, so you can do it systematically. And here's the firing rate of the neuron. You find this linear relationship. As you increase it, as the stimulus moves in the preferred direction of the cell, the cell responds more strongly when you have a stronger motion signal and sort of has this weak inhibition in the null direction. All right? This is what Guérin and Ries found if you do this fMRI signal and you plot it, you plot the percent change. See here the signals, 0.1%, very small change, but systematically in fMRI. And so we said, well, that's easy. There's a beautiful linear relationship between fMRI and firing rate. Okay, the referees loved it, immediately got in. Now, of course, and we were even so cheeky to assign a number, the ratio of this slope to this slope. All right, now, of course, the reality turns out to be far, far more complicated than that. Yes, it is true that on a simple task like this, where you're primarily a feed-forward, a stimulus-driven event, so you show something up, you flash something in the visual field, excites lots of neurons, they fire vigorously. In that case, you get this nice linear relationship between fMRI and single cell, and people love it. But now people do much more complicated experiments. So the best person who does this, again, is Nikos Logotitis. What he does, he has a monkey magnet, 4T and 7T, and he equips the monkey magnet with microelectrodes. Now you have to remember, you're pulsing, you're in this very strong magnetic field that will induce, of course, currents if you move anything. You've got these gradients and you've got radio waves. This is the worst possible environment to do very, very fine electrical measurements. So what he has to do all this very fancy shielding, lots of electronics to shield the microelectrodes that are sensitive. They're trying to pick up the tiny, almost imperceptible, chatter of the individual neurons among all this electrical racket that goes on in the magnet. Plus, of course, it's very, very, very loud. How many of you have ever been imaged? All right, okay, so it's, right, you get these very loud signals. All right, so maybe, all right, so what they did here, this is it, so you can do simultaneously in the behaving monkey, in the magnet, you get the monkey to behave, so in this case, fixates. You're recording the single neurons, you're recording the local field, and you're also recording the fMRI signal all at the same time. And this is sort of really only for the experts. So what you can see here, this flicker, this rotating flicker that goes on or off. You can see here, this is the fMRI signal over time. So this is 2 minutes, very long, very slow. This is in units of signal to noise. So you can see here, it's signal to noise is only 1. But you get these very reliable, very routine increase in hemodynamic, decrease in ball activity. In this particular slice here of this very high resolution of primary visual cortex. Here you get the different electrical signals. So they pick up all sorts of electrical signals from spikes. They're here called multi-unit activity. So this is specifically spikes to gamma oscillations. So this is 65 to 120 hertz, 125 to 300 hertz. A lower frequency, this is 20 to 60 hertz. This is conventional gamma, alpha, theta waves, and very slow delta waves. Those you would get, for example, in deep sleep. And they can now correlate very systematically what the relationship is between spikes or local fields and fMRI. And it turns out the upshot, so by the way, notice here the signal to noise. Here the signal to noise is 1 for fMRI. For single units, or for gamma, etc., it's 20. So electrical signals are much, much easier there to pick up, partly because, of course, you're sticking an electrode directly in the tissue. So this is one case where, for example, they found discrepancy. So here you have the stimulus that comes on, that stays on here for 14 seconds. You can see the spiking activity, multi-unit activity, gives rise, of course, immediately neurons. When the stimulus comes on, the neurons will fire very vigorously, but then the signal dies away after maybe a second, and then here you just have noise. While the underlying bolt signal, stays on for much, much longer, because once again, the bolt sort of is the collective change in hemodynamics following this stimulus. If you're looking at high gamma oscillations, 20 to 60 hertz, you can see the signal here tracks somewhat more closely fMRI. So the general conclusion is that local field potentials, particularly in the range 20 to 60 or 10 to 60, correlates much better with the fMRI signal than individual spikes. And there can be significant discrepancy, and this is the last slide, end my talk with this, because sometimes these are very interesting when you do not get a relationship between single spikes and fMRI. So the experiment's a complicated one. This is done by David Leopold at NIH, but essentially involves one of these experiments where they manipulate, I'll talk more about them in a couple of weeks, where you manipulate the relationship between what's on the retina and what you see. So essentially there are all these masking techniques developed for humans where you're looking at something but not seeing it. For example, you're looking at something in one eye, in the other eye, the corresponding location, I flash various stimuli. All you see is the stimulus in the right eye, but the stimulus on the left eye is never visible, although it's present on the eye. And then you remove the right stimulus, and then you immediately see what's on the left. So this you can get at awareness or at consciousness, because now you are studying. In both cases, you have the same physical input. Sometimes you see it, sometimes not. And David Leopold did this in this Nature Neuroscience paper, or Nature paper. He did this for monkeys in primary visual cortex. And what he sees is, and he gets a monkey to tell him what the monkey is seeing. It's really beautiful, whether he's seeing the stimulus or not seeing the stimulus. And he's essentially asking the question, so here what he plots in different signals. So this is the bold fMRI signal. This is the local field signal in this 5 to 30 frequency range, or high gamma frequency range, and these are spikes. Here he plots the perceptual suppression index. So this essentially, think of it, how much the signal corresponds to what the monkey is seeing, which under these conditions, it's a so-called perceptual suppressed condition. The monkey shouldn't be seeing anything. Should not be seeing this stimulus, although it's present on his eye, but because of the masking, the monkey tells you, I'm not seeing it. And what this tells you, that the fMRI closely follows what the monkey is seeing. And people have done many of these experiments published. They were very popular in Science and Nature like 8 years ago, where the people did these sort of experiments using binocular rivalry and other form of masking to show that in primary visual cortex, the fMRI bold signals closely follows what people see. If they see it, the signal is high. If they don't see it, the signal is low. This bugged me because Francis Crick and we wrote a paper in Nature 1995 saying exactly the opposite, that primary visual cortex does not have access to consciousness. In other words, the neurons in primary visual cortex do not follow the conscious percept. And that's exactly what this says. Right, the spikes that you record, particularly from whatever cells you record from, which tend to be large, do not follow this suppression. So they merely fire on. They are unperturbed in primary visual cortex by the fact that the animal doesn't see it. They essentially signal what's present on the retina. Of course, there have to be neurons higher up that signal what the animal sees, but it's not in V1. So, and we now understand better these discrepancy between what you get with spiking and bold. So the bottom line is that fMRI signals primarily synaptic activity, particularly synaptic input. This synaptic activity might be vetoed by other neurons. So think about, this has been shown beautifully in the case of cerebellum, but think about what happens. You have a stimulus that comes in, the input fires vigorously, and then there's an inhibition that also fires vigorously. The net result is the principal cells don't fire, but there's lots of activity, right? There's all this presynaptic churning. Glutamate is being released, GABA is being released, it needs to be replenished, requires glutamate. All that signaling shows up in the fMRI, even though the principal neurons are not firing because they're shut down. So in other words, the output to that area is shut down. So what fMRI tells you, if you get this blob of activity in area X, for sure what it tells you that area X is very active right now. There's lots of synaptic input, lots of local processing. It may not tell you whether or not area X actually is active and sends the output to other areas. That you can tell. In some cases, like simple motion stimulus, when there's no masking, it's true that the output neurons fire. In other cases, like this experiment, the output may not fire. And so this is what people now starting to think about and trying to make models. Sort of, this is the canonical microcircuit of Kevin Martin and Rodney Douglas, where you have this supragranular and granular cells in layer 4 and layer 2, 3, and layer 5, layer 6 in GABA. So the thinking goes that you can have an overall regulation that if your neurons and they're balanced, so you roughly have as much excitation as inhibition, but here, you double your excitation, you double inhibition. Here, you're really going to get a very strong fMRI response. Although the difference between the two isn't very large, but there's so much more spiking particular presynaptic input going on than in other conditions. So people are now trying to make quantitative models to try to understand much better the link between fMRI and the underlying neural signals that we mainly care about. So with that, I'd like to thank you for your attention. Well, I mean, there are all sorts of technological artifacts. Jung-Shimming, magnetic field homogeneity. As I said, it's really particularly bad once you get closer to the ventricles. There are lots of statistical artifacts. Like with any techniques, people are slowly coming to grips with them over the last couple of years. These statistical artifacts. What will, however, remain for a long time is trying to. So we care about neural mechanisms because what we see are hundreds of different cell types. And and of course you don't see that in the fMRI, right? fMRI, you get this global collective response. The way I think about it is like, imagine you're up in space and you're trying to track people, but you don't have the technology. But what you do have, you can track the power consumption on the grid. So you can see in the evening, they move from downtown to suburbs. In the summer, the power consumption goes up because of air conditioning, etc. But you're unable, for example, to track all women, all men, all policemen, all kids, right? Because you don't have tracers. And what we're doing here, we're getting individual tracers to track those individual subpopulations. So I think that's the biggest problem with fMRI. It's a fantastic technique because it gives you whole brain coverage, right? It tells you in the entire brain where things go on. Well, it has much higher signal to noise, has less spatial resolution, and you can only do it a few times, on each particular subject because of how much radiation the subject is exposed to. So I know very few people who it's still used somewhat in clinical domain. It's still used for specialist application, but it's really fallen dramatically out of favor. The question is, are people somehow using some new technique and tools to directly image electrical activity? I don't know of any. I don't think NMR can be exploited to do that, because essentially you would require that the electrical field has to have an effect on the magnetic resonance. And the fields here, given the time constant, etc., I don't think that's going to happen. That's why people use EEG, but of course EEG has the same problem. You have this inverse problem, right? You're outside here, and what you're seeing is the collective results of electrical activity in millions of neurons and other cells, and so you can't pick out individual, very localized activity. No, I mean, for that, I think you have to open the skull and put electrodes in, which is not so good for people. All right, thank you very much.